Okay, let's start. So good morning, afternoon, evening, hola, hello, bonjour, hola, ni hao, konnichiwa, 안녕하세요, and hello all, and welcome and thank you, SynBYSS people, for joining us the 82nd seminar, already 82nd, and I call it on UC Santa Barbara Day. I just got back to home after the SynBYSS 2023 conference in Minnesota. It was great to meet many synthetic biologists and share my research there by giving a talk and then discussing with many amazing people over there. And starting this coming Monday, I will have 1 month non-stop seminar again and conference trips without coming home. And I look forward to seeing many of you at conferences, including SEED, EBRC, and also Metabolic Engineering 15 in Singapore, where I'm chairing over there. So, okay, it is my tremendous honor to introduce very briefly, I'm not sure I can do so, but I will try, our pioneer speaker, Professor Michelle O'Malley. She is a chemical engineering professor at UC Santa Barbara and an associate director of UC Santa Barbara bioengineering program. I believe she just appointed very recently. Congrats again. She is a leader in microbiome research with the pioneering paper published in Science with her former postdoc and my academic brother, Kevin Solomon. More importantly, she is a working mom of many children. I heard recently her children are a bit sick, and then the challenge, I know how many challenges she have as a working mom. And also happy Mother's Day, a little bit, days ago, although it's not today. I always feel all moms are Wonder Woman and working moms are super Wonder Woman. So Michelle, I truly thank you for your time and for your contribution to the scientific society and advising Kevin Solomon, and also being a mom. Also, I thank you so much for your wonderful contribution to the community, especially nurturing young people, and virtual podium is all yours now. Thank you.
Great. Thanks so much, Taesuk. And it's really my pleasure to be here. I guess bridging from what Taesuk has said, I'm happy to be at UCSB. It's a lovely place, and I'm in fact outside. Most of the reason why I'm outside is not because it's a lovely place, but in fact because the power in my house just went out about 10 minutes ago. So I am coming to you live from Starbucks. I suppose this will be on YouTube, so hopefully not too much of an ad, but we all run on coffee as well, especially working moms. So I will put that plug in there. So I'm delighted to give you guys a little bit of an overview into some of the things that my group does at UCSB, and I will just share my screen. Apologies for the road noise in the background. But what my group does and is continually is motivated by and just inspired by is all of these weird microbes that are hanging out in nature. So I like to describe that a lot of what we do is engineering biology with these so-called weird microbes and microbial communities. And there's a picture of our lovely, beautiful campus there, and our lab is tucked over here. And it's certainly, if anybody is interested in joining us, either as a graduate student or scholar or a project scientist, I'm happily recruiting at all times. And so I can motivate some of what we're doing with our favorite emoji. So we do a lot of investigation in sort of weird microbial communities. And one of these is actually fecal materials. It's no secret that a lot of companies have really formed around the space of using these materials as even diagnostics or readouts of what's going on with gut health. But what I would argue is this is really a biotechnology resource, which probably sounds super strange. And part of the reason why is because in the gut and fecal materials, there is a wealth of beautiful diversity of microorganisms that divide and conquer pretty difficult tasks. And so what we're looking at is actually a breakdown or kind of a family tree, if you will, of microorganisms that are highly specialized in the role of biomass breakdown. This is from a paper that was published in Nature Biotechnology a few years ago now, but it's looking at the cow rumen. And the names in Latin are really not important, but what's more important is that you see that part of what is in the cow rumen in this case is not just one type of microorganism, but hundreds, thousands even, and they're all specialized to help each other. And so, so part of how I want to frame what we do is that we're looking at microbial communities from the standpoint and really the engineer's lens, that they're there as a team where they're dividing and conquering pretty difficult tasks. But as in any community, there are microbes that have friendships, so some sort of dual benefit. They're feeding off of each other, maybe cross-feeding some nutrients. They need to be around each other. There's also microbes that are competing for resources, might be enemies. And then there's microbes that will change how they feel about each other depending on the environment changing. So frenemies is how I term them, and everything in between. So these social dynamics really dictate, if you will, what happens in a community. From our standpoint, we're super interested in answering two seemingly simple sounding questions. The first is just who's there in these weird communities, and that's usually answered by sequencing and to some extent cultivation. The harder question is, given who's there, what is the microbe or population of microbes doing, and that is a much harder thing to address, and something that the field is really struggling with, and that we hope to, in some small part, try and answer. And this gives us a couple of opportunities. So one is kind of a really lofty goal. So given what we learned from nature's microbial communities, or the communities in the cow rumen, or in soil, or in sea sediment, right, where there's fantastic chemistry happening, can we learn from nature and nature's beautiful, diverse microbial communities to design communities synthetically that enable us to do whatever we want? So that might be cleaning up an oil spill and turning that into a nutrient, right? Or more practically speaking, taking plant biomass and using the enzymes in a diverse community to route and unlock the carbon contained in plant waste to a value-added product like a fuel, a chemical, or even a material. But this is still awfully hard because we as engineers don't really have a great handle on the design rules that nature uses to put these associations in place, and moreover, how to predict them, right? What I would also argue is that on this path, there's also something that we should pay attention to, and that is just recognizing the parts list of these microbial communities themselves. And so when I say parts, truly from a synthetic biology standpoint, this might be, recognizing that certain microbes that we isolate could be valuable chassis systems in and of themselves. We can use them for biomanufacturing or use them for bioremediation, right? But also that their enzymes, metabolic pathways, and transporters and biosynthetic capabilities can also be ported to microbes that we do understand, right? So yeast, E. coli, even mammalian cells, right? And so this is a snapshot of just part of what my lab does. So we've gotten into this field of weird microorganisms primarily by studying an isolated anaerobic fungi that, as this cartoon illustrates, seek and destroy plant biomass. And they do so by producing really valuable enzymes. And so Kevin Solomon, that Taysuk mentioned earlier on in the talk, was the first postdoc in my group, and really was responsible for cataloging for the first time this comprehensive parts list of enzymes that were in these fungi. And that has led to a lot of really interesting areas for us and others to explore, specifically looking at how the enzymes that these fungi are actually held together. Many of them are scaffolded in fungal cellulosome complexes. So we have tried to learn the design rules of how fungi hold these together and then make synthetic complexes inspired by them. We've also gotten really interested in the membrane sensing capabilities of weird microorganisms, to the point where we have discovered and engineered some sugar transporters, again, sourced from these anaerobes. And of course, can't forget about mixing and matching these microorganisms in anaerobic cultures. So we've tried to, again, learn and decipher these design rules so that we can route carbon where we want. This is kind of what I like to call the tenure slide. And then since I've gotten tenure, my lab has exploded in application space. So we've gotten a little bit more into other areas. So bridging into how microbes fight with each other actually is a route to discovering natural product synthesis, that we are working with some collaborators to even explore biomaterial-based capabilities out of these anaerobes, looking more at imaging and structural biology that feeds into some of the more ocean-centric work that we've done with some collaborators at UCSB, looking at diatom engineering from a material standpoint, and even how microbes are associating with plastic in the Santa Barbara Channel. And so that's a little quick introduction into some of the things that my group has done, and just want to give a shout out to everyone in my group, past and present, who have made all of this happen, because I'm not useful in the lab anymore, but I try to help them on their path. And on that spirit, I would, I'm really happy to turn the floor over to Taesuk and Neil, because I know he's got a lot of really exciting stuff to tell us about. And again, my great honor to be here, and I'm happy to stick around.
Oh, this is our honor to have you. Thank you so much. It's brilliant talk. You know what? I believe future is basically now, related to research on microbiome. So that's the reason I also chairing or organizing a conference, microbiome conference I think in December. So I hope many people joining the conference in December in Berkeley. So thank you so much. I mean, absolutely agree with you. You're doing fantastic work, and then especially nurturing future leaders, including Kevin Solomon. That is absolutely my favorite part you already done. So thank you so much for your contribution.
Thanks, Taesuk.
Yep. Okay. So now the main speaker of today, with a longer introduction. Dr. Neil Dalvie is from San Diego, California. And interestingly, I met him for the first time in another country, Portugal, in a conference. And at the time, we had great time having dinner and then discussing many things, including science and life. He attended Northwestern University, where he worked with Professor Josh Nenner, another my colleague, and doing fantastic job, and was inspired by the intersection of chemical engineering and biology. Neil did his PhD at MIT with the Professor Chris Love, where he worked to develop yeast as an alternative host for manufacturing therapeutic proteins, including several vaccine candidates for COVID-19 that reached the clinic. I believe he will talk about that interesting, exciting data today. Interestingly, Michelle, Neil, and I are all MIT alums, if I remember correctly. And now he is a research fellow at the Synthetic Biology Hive at Harvard Medical School, working with Professors Pam Silver and Mike Springer on secretion of complex biomolecules with applications in therapeutics, CO2 capture, and precious metal recovery, all important topics right now and in the future. Neil envisions an academic career in chemical engineering and is excited to connect about all things in synthetic biology. I truly believe he is true rising star, and if you considering hiring future leaders in near future, you should consider Neil very seriously because he will snapped by multiple university if you delay. So Neil, thanks so much for your time today. And please take it away. It's all yours now. Thank you.
Thanks, Tasek. Thanks, Michelle. It's exciting to be here with you guys, both leaders I look up to. So as Tasek said, I finished my PhD last year at MIT in Chemie. So this talk is focused on a portion of my work there. And now I'm at Harvard Medical School. I have one slide on my current position later, but I won't focus on that. Hopefully, I can give more talks about that in the future. So in grad school, I worked on developing low-cost manufacturing processes for therapeutic proteins mainly, and we used yeast for that, and I'll talk about why. And today I've focused on one case study of that in development of a vaccine candidate for COVID, among other things we worked on. So this problem with the pandemic is very easy to motivate from a manufacturing lens. I like to start with this number, that the number of vaccine doses that were manufactured globally for all diseases before the pandemic was 5 billion. So 5 billion doses per year of vaccines for the whole world. And then the pandemic comes and suddenly we have a need for more than double that to just address COVID-19. So it's kind of a step change in manufacturing requirement globally. So this is obviously a large technical challenge. To add to that challenge, nonprofits like World Health Organization and Gates Foundation, they know where these vaccines are needed most and where they're less likely to get. And so they set cost targets that to reach patients in low and middle income countries, we need to be manufacturing and selling these vaccines for ideally less than $3 a dose. To really reach some of the most rural areas of the world, that target is actually much lower and needs to be at cents per dose. To give a sense of the scale here, we're talking about vaccines, which are actually some of the more manufacturable types of drugs. If we look at the access to more complex therapeutics like monoclonal antibodies, this problem is even worse. So currently, 80% of all mAbs are distributed and deployed in just the US, Canada, and Europe. So access to high-tech therapeutics is a big challenge. So what does it mean to manufacture something? This is my very simplified process diagram, kind of gets at some of the main points. So we take some raw materials, we feed those into usually a cell culture called the upstream part of the process. We get some amount of our product out. We can quantify that with a titer. Then we have to purify that product. And then based on the yield of that purification process, we get maybe a volumetric productivity. That's how much can we make per day. The separation here is really a key part of the process and is where a lot of costs come from, mainly because making things in cells is complicated. Cells make a lot of proteins that aren't your protein, so you have to separate the product from those proteins. And what's even more challenging is if the cells make some version of your protein that's not quite right. That can be very expensive to separate from the actual product. It is important for not only purity, but like safety. These things are expensive to operate, so facilities cost a lot of money. The range here is big. Some facilities are small, some facilities are very big. But in any sense, it takes a lot of years to construct facilities. They don't last forever. And so the capital expenditure for biomanufacturing is very high. So if we want low-cost manufacturing, there's a couple keys that we want. First, we want high upstream titers. We want our cells to be making a lot of product. That's just direct cost savings. The more material our cells can produce per volume. We ideally want that product to be secreted. It's very challenging to crack cells open and purify the product out of the goop inside. But if they spit it outside for us, we only have to purify it out of the aqueous process fluid. It's much easier. We want that product to have consistent quality. So if there are variants that are difficult to separate, that's going to add a lot of cost. And then finally, especially for something like a pandemic, we want this process to be compatible with existing facilities. So we don't have time, if there's a new virus, to construct a facility to make a vaccine. We need to deploy a manufacturing process in a facility that already exists. So our solution to this at the Love Lab at MIT is to use microorganisms. Specifically, we like this yeast called Commagatella fafai or Pichia pastoris. Those are equivalent names. This yeast has a couple great things about it. It can be grown to very high cell densities, which gives you a very high volumetric productivity. It can secrete proteins into the extracellular fluid, so that's really nice for purification. And then it has some nice genetic process control, so we can grow the cells up before they start making the product and then turn on production of the product by changing what we're feeding them. So we'll talk more about that later. That makes it nice for process operation. And then maybe most importantly, especially from the lens of the pandemic, is this yeast is already deployed at very large scales around the world. So smaller proteins like insulin or vaccines for hepatitis B are produced at very large scale at very low costs in countries that we might want to target for low-cost manufacturing processes like India. So because of all these reasons, we think that K. fafai or Pichia is a nice host for kind of rapid development of low-cost manufacturing processes. The way this manifests itself in the lab is because yeast grows so quickly, we can iterate really fast in the lab on either different process conditions or different product sequences or different manufacturing conditions. So we can quickly iterate and engineer products and yeast strains to fit our purpose. And then also yeast biology is very simple compared to the biology of other manufacturing hosts like mammalian cells, for example. And so we can actually deploy kind of newer high-tech sequencing techniques to look inside the cells and see what's going on in our manufacturing process. And we can do that a lot cheaper and more high throughput than we can with a more complicated host. So now turning to COVID, so we were kind of working on this with a few different products, and then the pandemic happened, and we were funded at the time by the Gates Foundation, who essentially at that time diverted all their resources and said, you're already working for us. What can you come up with for a COVID vaccine? So we looked at the problem same way a lot of groups did. The obvious vaccine candidate for COVID-19 is the spike protein that decorates the surface of the virus. This protein is very large to make as a protein drug. It's actually easier to make as an mRNA drug, which most of us know. If we were to make the protein, it's a trimer that's almost 400 kilodaltons. So that's a sizable, and the bigger the protein, generally, the harder it is to manufacture it. We and others identified a smaller chunk of this protein that might be also a viable candidate, and this is just the receptor binding domain, so kind of the business end of the spike protein, and it can be expressed as a subunit that's only about 25 kilodaltons, potentially much easier to manufacture. We relied on some work from some very insightful scientists from back when the SARS-CoV-1 virus had emerged, who figured out that you could actually express the RBD of SARS-CoV-1 in yeasts, including Pichia. And of course, now in hindsight, we know that the RBD is actually a good vaccine antigen. It's known that it can elicit potent neutralizing antibodies. So we got to work in the lab and we developed a draft manufacturing process. So this involves choosing what we're going to make. We clone a yeast strain that's going to express that protein, identify a high producing clone, develop a purification process from flask experiments, and then eventually perform an end-to-end production run on one of the custom bioreactor systems that we had at MIT. This system has been published on before. It's called Insight. It's kind of a perfusion benchtop scale reactor system. This technology has since been spun out into a company called Sunflower Therapeutics that's deploying this technology to make a broad range of protein drugs. But in the lab in 2020, we were able to go from just the idea of a sequence to actually 30 milligrams of preclinical product in less than a month, which is pretty cool. That product is ready to go into mouse experiments, for example, and kind of start the drug development process. For reference, I believe Moderna in their vaccine development around the same time did this amount of work in 21 days, and they had an entire company with at that time hundreds of people, where we did this with 4 people. So that's my humble grad school brag. So once we had RBD product and could start testing it, we then worked with several collaborators that I'll thank later, who helped us develop an actual vaccine drug product. So we imagined at the time, and now it's well known, that the RBD is a good vaccine candidate, but when it's decorated on some sort of particle multivalently, it can be an excellent vaccine candidate. This has now been tested and it's well known. So we teamed up with help of the Gates Foundation with a company in India called the Serum Institute, which is one of the largest vaccine manufacturers in the world. They had previous experience with Pichia, and they used yeast, for example, to make a hepatitis B vaccine that we could leverage. So with Serum, we designed a vaccine product where they take their hepatitis B nanoparticles that are made in different yeast called Hensenula, and in a separate tank, they would manufacture the RBD in Pichia, and then basically mix these together. And each product was decorated with one of these peptides, spy catcher, which is a slightly larger peptide, and spy tag, which is a slightly smaller peptide. Those are just genetically attached to the proteins, and when you mix them together, they spontaneously attach and they form this RBD nanoparticle. So we made some of that, both at MIT and in collaboration with Serum, and we did some early animal studies. These were done at the Baruch lab at HMS, which is where I am now, with a postdoc, Lisa Tostanovsky, who led these studies. So we injected the vaccine into monkeys, and we saw that with multiple combinations of adjuvants, they had elevated levels of neutralizing antibodies, and then upon challenge with SARS-CoV-2, reduced titers of viral RNA in both the lung and the nose. So this was pretty exciting preliminary data, enough actually to motivate Serum to move forward with a clinical program in India and elsewhere. So I'm going to actually spoil the end of this program before I talk about the most interesting stuff. But so with Serum, they went to the clinic in both Australia and in Zimbabwe, but with the vaccine that was developed at MIT. So this clinical trial went 1 to 2 years. It's currently on a business hold and is probably not going to see a phase 3 or 4 trial. The main reason for that is a business hold, and primarily a very similar vaccine from another manufacturer in India was approved. So, that's ultimately great news for the field. The vaccine that was approved is from a company called Biological E. Their vaccine is called Corbevax. It's based on the RBD, and it's now been deployed in millions of people. So pretty exciting for Pichia, for the RBD, and a lot of success for the field. There's actually been RBD-based vaccines approved all around the world now. So there's one from Cuba, there's the one from India, there's one that's came out of the University of Washington that's manufactured in Korea. That's actually an RBD nanoparticle, and it's been shown to be highly effective. So this ended up being a real technology, which is kind of cool. So while the clinical program was getting going, in the Love Lab, we then said, okay, we can make this thing in yeast. It's not optimal. So we set our sights on improving the manufacturing process and ultimately even the product sequence of this vaccine. So I'll talk about kind of 3 little stories about how we did that. We deployed several techniques for this. So this was kind of the bread and butter engineering processes that we were developing, where we're going to look at the quality of the product, but also look at the health of the host while it's making that product, and use that to inform both sequence changes to the product, and then also changes to the yeast host. So first we looked at the quality of the RBD, and when we make it at lab scale and once it was scaled up, there's kind of one critical quality attribute of this molecule that sticks out, and that's that it tends to aggregate. So we can see it high on this gel, where there's a high mass glob of RBD that still comes out of our purification process. This is exactly the type of product variant that's very expensive to separate from the real product and can pose safety hazards if you don't do that. So the best way to eliminate something like this is not actually to purify it out, but go way upstream and make our process so that it doesn't have it. And the farthest upstream we can go is the molecule itself. So with my fellow grad student, Sergio Rodriguez, we analyzed the RBD and kind of looked for reasons why this molecule might aggregate to itself. And Sergio noticed that there were these hydrophobic hotspots on the molecule. So patches of protein surface that might glob onto itself. And the main hotspot was actually in the ACE2 binding pocket. This is the receptor that the virus binds to enter cells, and this is the part of the molecule that we want neutralizing antibodies against. So it's difficult to think about mutating this region of the molecule without breaking the function of the vaccine. So we did this in an informed way. So we looked at relatives of SARS-CoV-2 and found mutations that might conserve the structure and function of the molecule, but alleviate this hydrophobic patch. So we made a couple different mutations and screened them, and I'll show you the winner. But our engineered version of the RBD, which has just 2 amino acid mutations, first, it can be manufactured much better than the original one. So the yeast simply make more of it. It's a more well-behaved molecule for secretion from cells. Second, it seems to have less aggregation. So this is a size exclusion chromatogram, and on the bottom in red, we can see the peak on the left is greatly reduced. So we're seeing less aggregated protein. And if we look at the intrinsic tendency of the molecule to aggregate by light scattering, we see that this molecule tends to kind of start falling apart and aggregates at a higher temperature. So it's more stable, even thermodynamically. We then had to check to make sure that this molecule is still a vaccine antigen. And so first we looked at the binding to the ACE2 receptor and some known antibodies that can report on the function of the vaccine. And interestingly, not only did it retain binding to these molecules, but the binding was actually improved. So something about our change is making the molecule bind more tightly and more stably to antibodies or the human receptor. When we tried this in a mouse study to see if it could elicit a neutralizing response, also surprisingly, we saw that the improved antigen actually had more immunogenicity in mice. So we see a better response to the vaccine in the mice than with the original molecule. Okay, so that's all really cool, and we had some very unexpected results from that we were actually able to improve the function of the vaccine. We reduced this critical aggregation a lot, but as you know, this gel, this is the engineered RBD, there's a lot less aggregation, but there's still some there. So we kind of then asked to go, even deeper into the primary sequence of this product variant and try to figure out why does this aggregation remain here. So in the lab, we then separated out that product variant just on a size exclusion column so we could perform mass spec on the correct antigen and then also the aggregated species. We can identify the masses and find that the aggregation actually has a larger mass than the original antigen. And when you figure out what these extensions are, it turns out to be the signal peptide. So when you secrete a protein, you have to put a signal peptide on it that tells the cell that that protein should be secreted. The best signal peptide in Pichia is called the alpha factor from Saccharomyces yeast. And what we're seeing in this aggregated product fraction is that the signal peptide is not fully cleaved off of the RBD. It is for most of the product. That's the correct mass fraction. But in the high mass fraction, we're seeing a high percentage of the species that has this signal peptide still hanging on. So how can we help the cells cut the signal peptide off more completely? One hypothesis was kind of already built in. So in our vaccine product, we had taken the RBD and added this small peptide called SpyTag. We had originally added this peptide to the C terminus of the molecule, not for any particular reason. So one hypothesis was, well, let's move the SpyTag to the N terminus of the RBD. The structure of the vaccine product should be the same, and maybe having the SpyTag stick off the N terminus of the molecule will help the cells cut off the signal peptide from the N terminus. Similarly, it's been known in the literature that in the native signal peptide system, there is a 4 amino acid sequence that can follow that signal peptide. Those residues are EA, that should help the first protease, KEX2, cut the signal peptide off. And then those 4 amino acids should get cut off by a second protease called ST13. So we also tried adding these 4 amino acids to help cut the signal peptide off. And sure enough, we find that when we do any of these interventions, the protein after purification is almost perfect and has no high molecular weight species. And in fact, only the version where we have SpyTag on the C terminus and nothing on the N terminus do we see the problematic high molecular weight species. Interestingly, this kind of phenomenon of making the N terminus more accessible also applies to the second protease, ST13. So if we put the EA residues, they are only cleaved by that second protease, ST13, when the SpyTag is on the N terminus. Finally, we showed that we could actually get this benefit from not just 4 amino acids, but even down to just 1 amino acid, and not just a glutamine, but also any number of kind of amino acids you might choose. So this data is a little bit unsurprising, but perhaps nice in its simplicity, that if you just add a glutamine, serine, alanine, pick your favorite simple amino acid, you can eliminate any sort of N-terminal problems. We also put some controls here. Proline doesn't work. That's maybe unsurprising. But the conclusion here is if you kind of wisely choose an amino acid to add to the N-terminus, you can really clean up your protein quality. Interestingly, we showed that this worked with some other proteins too. So we had worked on a vaccine product for rotavirus that's a similar subunit protein. It also has a peptide addition on the N-terminus. That's an epitope from tetanus toxin. And we performed a similar experiment with these two molecules and found similarly that only when there's nothing attached to the N-terminus do we see incomplete signal peptide cleavage and a high molecular weight variant. And it seems to be a generalizable phenomenon. So we can kind of draw this model that, you know, when the N-terminus of your protein is buried in a recombinant system, the signal peptide may be incompletely cleaved. Whereas when we add some sort of extension, even down to a single amino acid, we can properly process the N-terminus of the protein with actually 2 different proteases. Okay, so that was a lot of product sequence engineering. We also deployed some yeast genome engineering to kind of address this problem. And this is a, strain engineering for protein titer is a very big question. There are many ways you could approach this. The ultimate question is, why isn't the cell making more RBD than it is? So we chose to kind of go after this with RNA sequencing. So we compared cells that are making our COVID-19 RBD with cells that are actually making one of these rotavirus antigens that is secreted at very high titers. So we're looking for differences between the medium titer strain and the very high titer strain, see how we could further boost the productivity of the RBD. And what we see is that in the strain expressing the RBD, genes related to protein folding stress, and protein degradation are upregulated compared to the high titer strain. So we might hypothesize that instead of being secreted, the protein is actually, kind of being translocated, and then somewhere during the folding process, it's being degraded instead of making it outside the cell. So this is kind of great biology, and we can see the problem here. Unfortunately, it doesn't really tell you what to do about this. There are many ways you could approach this. We took one strategy that really worked here, and that was to look at the induction of the recombinant gene. So in Pichia, the recombinant gene is induced by growth on methanol. So we first grow on glycerol, and then when you put methanol in, it activates an inducible promoter and makes the RBD. So we thought, well, maybe we're putting too much RBD in. And when we express too much of the gene, we kind of overwhelm the protein folding machinery, and cells end up degrading most of the product. So very simply, we took cells and we just fed them less methanol to try to induce less of the recombinant gene. And sure enough, the titer starts to go up when we feed them less methanol and maybe induce less of the recombinant transcript. We then made an engineering change to the genome where we activated the recombinant transcript, activated expression of the recombinant gene without methanol at all. So this was by integration of an extra native transcription factor. And we find that we can actually get this boost in productivity with no methanol now. So not only is the titer higher like we wanted, but we've also kind of now changed our process conditions where we can feed them only a non-repressive carbon source like sorbitol instead of feeding methanol at all. We checked the transcriptome of this new strain and find that, as we see in purple, the engineered strain now does not show this kind of stress protein degradation signature that we saw in the base strain making the RBD. So hopefully now we've fixed the problem that we saw earlier. And excitingly, when we ran this in our bioreactor system, this is the product of the bioreactor over time, we actually see that this new strain is much more process friendly because it doesn't need the methanol feed. So methanol is an oxidatively expensive carbon source, and when we cultivate for up to a week in our bioreactor, we see a lot of lysis in the top gel, and kind of the product is petering out, the cells are not looking healthy, whereas in the row on the bottom, in the methanol-free condition, we get nice, consistent production of the product for up to a week. So I've described some changes to the strains and then also changes to the engineered antigen. We transferred all of these technologies to Serum Institute during kind of their clinical productions. Interestingly, the methanol engineered strain also kind of made a qualitative change for Serum and allowed them to scale up to a bioreactor scale that they weren't otherwise licensed to scale up to with a flammable feed like methanol. So because of our engineering changes, they could scale up pretty quickly to actually 1,200 liters. And I'm showing here kind of consistent product production between our flask material and the material from Serum at 1,200 liter scale. And to kind of drive this home for the real process nerds in the room, this is the intact mass of the proteins compared from the MIT product and the Serum product. What we're seeing in purple is the glycan envelope. So yeast will monosylate the product. It's not really relevant for the function of the vaccine, but we can see just striking similarity in even the abundance of the different glycosylated species between our process and their process. So this is pretty amazing scale-up result that the consistency we get in a flask and at huge scale is so good. Okay, so main takeaways from all this, a lot of these engineering changes and our success in the clinic were driven by use of the microbial host, specifically Pichia. These can just be grown at so much higher speed and at lower costs than other hosts like mammalian cells. We saw that small changes to the product sequence or actually just the vector design can lead to big improvements in the quality and titer of the protein we make. And obviously it's best to implement these changes as early as possible in process development, even during discovery and design of a product. We should be considering the manufacturing. And then finally, I gave an example of some host engineering. Many synthetic biology efforts to improve manufacturing hosts kind of use high throughput techniques and multiplex many changes at a time in order to see benefits. We showed an example here where it's kind of a qualitative change to how the host operated that interacts with the process operation and kind of gave like a big step change in what the host could be used for and also the titers we gave. So this work was made possible by many people. These were large team efforts. The vaccine work primarily was funded by the Gates Foundation, a program called Ultra, based out of the Love Lab at MIT. And a lot of the development of Pichia in general as a host is happening in a consortium called AltHost that's run out of the Love Lab to develop Pichia and other alternative hosts for making not only vaccines, but also therapeutic proteins that are more complex like antibodies. Just update on me, I'm now at Harvard Medical School in the synthetic biology hive. This is a new center founded by Pam Silvers, one of the thought leaders in synthetic biology in general. We're thinking about kind of global challenges related to health, but also related to climate and food security. And so really new things. I'm leading a project to use microbes for mineral dissolution primarily. So it's been a learning curve, but in the long run, I think there's a lot of potential to use kind of the precision tools we use in pharmaceuticals for even bigger scale processes related to climate and food. And going the other way, that the scale that we'll need to address problems for climate change and food security can hopefully inform how to make truly commodity medicines. So thanks for everyone's time, and I'm happy to take questions.
Amazing. I love all your work, also including the last slide. That is actually a very important topic we should address in the future, in near future, I believe. So thank you so much. Michelle, do you have any comment or question?
I mean, I'm pretty familiar with Neil's work, but I mean, I think what I would invite Neil to do, since he's told us a story that's so scientifically rich and compelling and touches all of us in terms of vaccine development, right? What advice do you have for people who are joining research labs and looking at synthetic biology as a field, if they want to follow that path, or how were you kind of drawn in, and how did that feed your passion and your scientific direction? Can you maybe, hopefully that makes sense.
Yeah, I think so when I was an undergraduate, I got inspired by synthetic biology, and we were making promoters for GFP, for instance, looking at dynamics, very fundamental SynBYSS project. And then I showed up at MIT and I was looking to do similar stuff. And I think synthetic biologists often run into a problem where you can come up with very cool experiments to do that are satisfying and use the latest and greatest technology, and sometimes you have a hammer and no nail. And these maybe aren't actually the best solution to some sort of problem. So I think what led to success for us, and the pandemic didn't happen for several years. My PhD kind of screwed around a little bit first, but the key, I think, is to solve the problem, not use your favorite tool. And this is a very engineering mindset that I now have, and that's different than, it's obviously important to do fundamental biology, but I think we need more true biological engineers who are looking at problems and trying to find the simplest way to solve it. Mm-hmm. So I think that was some of the biggest learning for me in grad school, was to let go of what I thought could be the coolest experiment for us to do, and instead kind of like get on board and engage with either the companies we were working with or the Gates Foundation and actually understand what was a problem for them. And this yeast strain that doesn't need methanol, I just, it's not flashy biology science. It's very simple genetic change, but I love that story because it solved such an easy problem for the people we were working with.
Terrific. I think that's wonderful advice for everybody watching, including the senior people too. I think it's good reminders for all of us, right? That it's
Yeah,
not about the
absolutely.
flashiest thing. It's about solving real problems, right?
It's hard. I mean, I'm just as prone as the next person to just, nerd out about some cool experiment we could do. So it's about finding balance there, I think. Yeah.
You know what? You realize that very simple but important thing earlier than myself, because when I was postdoc, I believe complex circuit is better now to go in the future. So we claim the most complex circuit in the world at the time. There's a 4-input AND gate. And then after I start my position as a PI, now I completely changed my mindset, because we need to solve the problem. And to solve the problem, probably simply circuit might be better, because most of the problem do not require 4-input and then respond to 4-input. We may need 1 or maximum 2, and then we could solve the problem. So that's absolutely great realization, very early stage. And that's why, I really admire your work and yourself. So that's wonderful. So Michelle, do you have any other question or comment?
I mean, I have several, but I guess, I think in terms of your current work, Neil, I guess on that mindset of practical issues, it may be just because I think you're stepping in kind of to newer territory from a synthetic biology perspective, which is it's super awesome, and I'm love to see where it's going to go. So what are can you give us some examples of some of the kind of tangible issues that your research is addressing from like a SynBio perspective for people that are not really familiar with that kind of a more environmental aspect, which just, I think it would be great to get some more people excited about that direction.
Thank you, Michelle.
Yeah.
That's exactly the same question I want to ask.
Yeah, so that's the same question I want to ask too. But I'll give you the quick synopsis. But the hive in general is people think about a lot of different problems here. So I've kind of narrowed in on one thing to start with, and that is on this idea of ocean alkalinity enhancement via enhanced weathering. So I didn't know anything about this last year. But the rock cycle is how the Earth kind of naturally captures carbon in a very permanent way, and ultimately will correct for climate change, but in 200,000 years. So, we need to, one idea for CO2 sequestration is to actually speed that process up and get rock erosion essentially to happen faster. So there's some recent data in the last 2-3 years that microbes can do this by a number of methods. They can basically acquire nutrients from a mineral that's in solution with them. They do this in one way by secreting an active biomolecule, which then can interact with the surface of the rock and help solubilize the iron, for example, that comes out of it. So that's what drew me into this project, was because I'm kind of interested in this, low-level lab assays for secretion and how you can measure that, which is actually not that far from what I was doing in grad school, day to day. So that's what kind of brought me in. But then, of course, the challenge is what is meaningful to do here? What could this technology look like? We're fortunate to collaborate actually with one kind of research organization called Project Vesta, and then also a smaller startup that's working with them that hopes to make technology like this, so we can, in parallel to what we're doing, kind of think about what this process might look like. And basically, the way to think about it is kind of like wastewater scale process for microbial fermentation on mining waste, for example, where the product is actually just alkaline water that you're going to dump back in the ocean.
Mm-hmm.
So it's pretty new lab experiments. So a lot of my task over the last 6-7 months has been to kind of figure out what can we measure in the lab that's relevant here, before we even get to any, fancy genetics that I'm sure eventually will be applicable to this. So we've been kind of just developing ways to measure mineral dissolution and carbon capture at lab scale that, working with our collaborators, we hope will be relevant when they scale it up. That's kind of what we've been thinking about. Yeah.
That's absolutely fascinating, because I'm thinking about not necessarily the same, approach, but for 10 years, I'm thinking about the metabolic engineering, because I'm a metabolic engineer by training as a PhD student, but metabolic engineering for 30 years basically use one feedstock typically, and one microbe, and one, product typically, and then one bioreactor. And why we limit ourselves to just that one situation, rather than, as you showed in the last slide, entire planet as a huge bioreactor. In that bioreactor, you could have your bacteria potentially reduced, greenhouse gases, and then some bacteria may fix nitrogen. I mean, that's natively happening, and then get rid of nitrogen fertilizer. At the same time, whatever bacteria from Michel's lab that could also do some cool conversion. In that way, we could now see the entire planet, soil and water as huge bioreactor that basically helping planet in a much more healthy place. That's kind of something crazy idea I thinking over 10 years, and finally last year I brave enough to put that in the paper. So I published in the Trend in Biotechnology, because one of the regular audience met the Trend in Biotechnology editor actually invited me to write about something. So I just decided to write about that one. So that is absolutely a wonderful idea, and I love that idea.
Yeah, it's interesting. It's so different than like making drugs, which happens in just like the most clean, controlled clonal way.
Right.
Where you know exactly what's in there, and you can, even if you engineer something that's like totally convoluted, like as long as it works in your clean tank, you can just do it over and over again. Whereas like in the extreme for like geoengineering, it's like, could you release a microbe into the environment?
Right.
And that's a complicated thing, and it's like there's a lot of societal implications to that, if you're doing this on purpose. I think that not only is it maybe risky, but it's also like impossible right now. Like if you put an engineered microbe in the ocean, it's gonna die. But so I think there's some starting point though where we can think about these processes in a tank. Like we have to start there. It's a very chemical engineering approach, but some sort of like semi-contained thing that you could use an engineered bug and start there. Interestingly, Michelle, we're thinking about, or our collaborators are really thinking about kind of like the ocean microbiome, and there's kind of like a secondary problem here that's actually even potentially bigger, more impactful. It's like, how could you take an engineered bug or bugs and reliably grow them in unfiltered seawater? I think is like a big interest, and in theory should be possible. You got to feed them something, and if you cleverly design the system, maybe your thing will win. But I think that's would unlock another level of scale for biology, to kind of use the lowest cost feedstocks.
Right. That's a very interesting point. Actually, my lab currently working on the same thing you mentioned, including we actually sample Mississippi River, and then wanted to test our engineered bugs in that Mississippi River, and also poop. Michelle's favorite, subject or material. We are also using Prof as kind of our media, and also at the same time also use the soil as real soil from the environment, and then see how environmental factor affect the viability of the engineered cell. At the same time, we heavily focus on biocontainment, because at the end of the day, we need to consider biocontainment issue when potentially we release our engineered bacteria into the the environment. But there are multiple challenging problems we need to solve. And then interestingly, three of us common interest is something dirty or waste, and then converting or using that on for something good, I guess. Michelle, do you have any other thing?
I don't think so, but I've got to run because my computer's about to run out of batteries.
Sure.
But
sure,
it's
sure.
been
Thank
a
you so
thrill.
much. Yeah. So let
Thanks,
me
guys.
close in
thanks,
that
Michelle.
case. So thank you so much, Michelle. So let me close. So thank all for joining and staying today. We'll meet again on June 4th. Oh my God, already June 4th. Thursday, the same time, the same Zoom link. We'll have Professor Doug Clark, a Dean of University of California, Berkeley, and Professor Sang-Moo Jung at Johns Hopkins University. As usual, the follow informal chat will occur without recording. Please stay here if you are interested in chatting with today's speaker and me. I will promote you to panelists who can speak and show your handsome and pretty faces if you wish. And thanks. I will stop recording. Thank you. Just give me 1 second.